In this video, we're going to summarize cell biology. We're going to look at ideas from DNA replication to photosynthesis, respiration, and movement. We're going to look at the purpose, the outline of the process, factors affecting the rate, additional notes, associated diagrams, and relevant structures and their adaptations for efficiency. Now, this summary sheet is great at having all the information from the course on one piece of paper. Okay, we'll start with DNA replication, mitosis, and the cell cycle. So the purpose of these is that they happen prior to cell division. They produce DNA that is identical to the original strand, especially in DNA replication. We can see that we need an exact copy of the DNA so that cells can function properly. Okay, that's the reason why we have DNA. It acts as the code for life, and we need to make a copy of that one so we can grow and repair. So the outline of the process, okay, we've got DNA replication, the steps here. So first of all, we have the DNA that is unwound by that helicase enzyme. And then we have the bonds that are broken between the bases A, T, C's, and G's. We then have the free nucleotides that are floating around the cell. They then attach themselves complementary to the free bases. Okay, so we have the leading and the lagging strand. So the leading strand is made continuously, and the lagging strand is where we have the Okazaki fragments that is in the 5' to 3' direction. We have a few different enzymes here involved. We've got the RNA primer, and then we've got the DNA ligase, which bonds the Okazaki fragments. Okay, so we just talked about the DNA replication. We're now going to look at mitosis. So mitosis undergoes a few stages for the cell division. We have IPMAT and C, so IPMAT. So we first of all have interphase. This is where the chromosomes are replicating. During prophase, the chromosomes are condensing. And during metaphase, chromosomes align at the equator or the middle of the cell with spindle fibers. Anaphase, we have chromatids that separate two opposite poles of the cell. And we have telophase is the next one, where chromosomes nucleus reforms, new cells form as a cell membrane pinches in. And then cytokinesis is when we have the splitting and two identical cells that are produced. Okay, these are what mitosis is used for. If we scroll down to the bottom, you can see we have a diagram for DNA replication. You may need to look at another diagram that is in a bit more detail and larger to see. Okay, so factors that affect the rate of both of these processes, okay, these are actually controlled by enzymes. So whatever affects enzymes is going to affect the rate of both of these processes. So temperature, pH, substrate or enzyme concentration, and inhibitors. Okay, so leading on from that, we're going to look at enzymes. So enzymes are a biological catalyst that speed up a reaction and lower the activation energy, or the amount of energy required for the reaction to start. Now we have a few different models. We've got the lock and key model and the induced fit model. Okay, we have an active site, which is that main part here where the substrate binds to and collides for the reaction to take place. We have the enzyme first of all with the active site and substrate. We have the enzyme substrate complex, which is the next step, and then we have the products being released from the active site. So there are factors that affect the rate, as we just said before. Okay, temperature, pH, coenzymes, minerals, enzyme inhibitors, and poisons that block the active site. Okay, just one thing about enzymes, they do operate at optimum conditions. Anything outside of this range will result in the enzyme not to function properly. Okay, you can think about enzymes operating at an optimum temperature. Anything higher than the temperature will denature the protein, causing the shape to change, so the substrate can no longer bind to the active site. Okay, so we've got relevant structures and the adaptations for efficiency down the bottom here. Okay, now have a look at those ones.